All right, so thank you for the welcome, Deb, and it's nice to see everybody here. My experiment was on Pinto Abalone postlarvae and the effect that diet and the morphology of the Radula has. In one moment, I need to turn off the chime for people trying to enter. And, okay, great. So an outline of my talk. First, I'll give you a brief introduction to abalone with the assumption that you know nothing about them. In other words, what the heck did you study, Lillian, and why do we care? Then we'll talk about the diet that I fed them, which was benthic diatoms. And in other words, abalones eat slime. The experiment was a diatom feeding trial where 6 diatoms were nominated to be the best, and I'll tell you which ones were and which one was definitely not. Then I'll transition into the radula, which is the abalone teeth. Whether they're terrifying or cute is in the eye of the beholder. Then some conclusions to draw it all together, and there will be time for questions afterward. So this is a Pinto abalone. This one is hatchery raised, which you can tell by the numbers glued onto it and the fact that its shell is not covered in growth, because in the hatcheries there's not a lot of other biotic growth to grow on the shells, but they will get that when they live in the wild. The most likely place you are to have seen abalone in modern times is as decoration. So a lot of people have just the whole shells in their houses as a decoration, or both traditionally in native cultures and in modern times, the mother of pearl is used as a decoration on jewelry and clothing. You may have seen abalone as food. They are very expensive now and not real abundant in terms of going out and fishing them yourself. And if you cannot get abalone, you can settle for nothing less than abalone-like shellfish. And humans, of course, are not the only ones that enjoy abalone. Sea otters do as well. This leads to some interesting wildlife conservation problems for people in California to solve between the endangered sea otters down there and the endangered abalones down there. If you're a scuba diver, you may have seen abalone in their native habitat, which are subtidal kelp forests. Depending on the species of abalone, they grow from near the low tide line to much deeper than that. So you won't see them if you're walking on the beach and looking in tide pools. They're below the lowest tide. They live in rocky habitats where they can both cling to the rocks for stability and also hide in cracks from predators. They require adequate currents, and crustose coralline algae is a cue that the habitat is good for them. So crustose coralline algae actually gives off a cue telling postlarvae or larvae swimming in the water that it's a good place to make their permanent home. And the abalones benefit the crustose coralline algae by keeping it clean and cleared of other seaweeds. And the coralline algae is this pink stuff that's encrusting this abalone's shell and then also encrusting some of the rock here. Abalones are sedentary, but they do aggregate to spawn. And the spawning begins with males and females in close proximity to each other. However, there's no deposition of sperm to the eggs or vice versa. Instead, both are released freely into the water to float and to encounter each other in order for fertilization to occur. Once fertilization occurs, the fertilized egg develops into a trochophore larvae for about 24 hours and then develops into a shelled free-swimming veliger larva. Abalones are marine snails, and they look very much like a classic snail shape in the veliger larvae here. And then this is the velum that allows the larvae to swim around. It's like a whole bunch of little cilia beating. Similar to that, they aren't technically cilia. After a week or more in the water column, depending on the abalone species and the water temperature, the larva finds cues from crustose coralline algae for a good place to settle. It drops the velum, which can actually swim off through the water column by itself without the larva, and then goes through metamorphosis, which involves its head twisting all the way around, and then it starts to live on its foot with the shell on top of it. It's interesting to note that larvae don't eat. They are living off of reserves from the fertilized egg. There's a lot of nutrients in the eggs. And then after settlement, post-larvae begin to eat diatoms and slime mucus off of rocks, but they can live off of egg reserves for a while. Juveniles are abalones that are big enough to start eating macroalgae or seaweed, but are not big enough or old enough to sexually reproduce. They're also defined by having one respiratory pore, which is the holes you see in the shells. And the number of these pores that is open on any abalone at a given time is an indicator of its species. There is overlap in the number of holes per species, but there's a range that each species has. Abalone range worldwide. They're most common in tropical and subtropical areas of the globe. On this map, the color coding is not individual species, it's simply ranges. And the abalone at highest latitude are in Alaska and the west coast of North America. That's the abalone that I studied. In New Zealand in the southern latitudes, but it's not as big of a latitude towards the pole as it is up here. There are about 60 species of abalone worldwide. In North America, there's only one species on the Gulf Coast, no species are on the East Coast, and we have several on the West Coast. The Pinto abalone is the one that I studied. It is a species of concern in Canada, in the US, whereas in Canada and the International Union of Conservation of Nature consider it endangered. There are some healthy populations in Alaska, and even a small subsistence and native fishery is allowed up there, at least as of when I did my research on that a few years ago. And the threaded abalone is a subspecies of Pinto that ranges further south into Baja California. Flat abalones are not well studied, and there's some taxonomic discussion about whether they truly are found in Washington state or whether they're simply further south, or if it was a hybrid that was found in Washington, because many taxonomists believe the only species in Washington is Pinto abalone. In California and Oregon, the red abalone, and down into Mexico, has stable populations, and there's even some recreational fishery in Northern California. You can catch them if you don't use scuba equipment. You have a bag limit per day that's very small, and the abalones must be big enough, and you have restrictions on what type of tools you use. The red abalone are the really big ones that are iconic in California. So if you see pictures of big shell piles, that's the red abalones. There are a few that are farmed, and that helps reduce pressure for people to poach them if they can buy farmed species instead. Black abalones and white abalones are endangered, and pink and green are species of concern in the US. The Pinto abalone, like I said, is the abalone that I worked with. This is a picture of a larger one that I worked with. They live in cold water, so that makes them grow slower, and they're also smaller than many other abalone species. So where did all the abalone go if all these species are endangered or threatened? A lot of it went to legal harvest back when we did not understand the reproductive cycle and thought that the amount we were harvesting was sustainable, or else simply didn't really consider environmentally sustainable levels when the laws were being made. And then also poaching. This is a big truckload of Pinto abalone that was confiscated in British Columbia. It's quite sad because the abalones really clump up together to spawn, so you can easily, if you are poaching, you could easily end up taking an entire area's worth of population because they all got together that one weekend to try and reproduce. In Washington state, there was a recreational fishery until 1994. There never was a commercial fishery. However, in the 26 years since the recreational fishery was closed, the populations have not rebounded on their own. They simply don't have enough density for reproduction to be successful. Again, with the sperm and the egg going out into the water and trying to meet each other, it simply isn't feasible for the sperm and egg, statistically unlikely for them to encounter each other. And so while the adult abalones are not necessarily being harmed by environmental conditions, they are dying of old age and loneliness and unable to reproduce. So Pinto abalone restoration is happening in Washington state. The Puget Sound Restoration Fund is the main player in this, with partnerships with Department of Fish and Wildlife and other entities. The goal is to make populations that are dense enough for reproduction to succeed. And the method is by setting juveniles loose into the wild. These are 10 monitoring restoration sites that they actively work with in the San Juan Islands. And the scuba divers are holding PVC tubes that abalones are in. So the PVC tubes were in the hatchery tanks. The juveniles crawled into them because they like to be sheltered and hidden inside of the tubes. And these scuba divers will anchor the tubes down, then come back later and collect the tubes after the abalones have crawled out. Pinto juvenile abalones are produced in the Puget Sound Restoration Fund hatchery. That is an involved process that begins with paramating wild broodstock. So they feed the broodstock, the adults, seaweed for several months in order to fatten them up and get their gonads developed, and then induce them to spawn. They're finicky and won't necessarily respond to cues to spawn. And that cue is specifically hydrogen peroxide. I'm not sure how anyone figured that out, but it often works, but doesn't always. Then each animal, once it starts emitting its gametes into the water, gets removed into a separate bucket so that the gametes are collected individually. And then small amounts of the sperm and eggs are combined so that each male and each female that spawned on that day have equal opportunity to mate with each other. That maintains genetic diversity in the restoration juveniles. And so each of those pair matings, all of the siblings have to be raised in a separate tank so we can keep track of which family they're from. And that means that if the abalones are growing slowly or many of them died, we can't simply combine them into one tank and then start breeding more for the next season. The abalones are always outplanted in spring and early summer. So if they aren't big enough for outplanting 1 year, they're going to wait a whole other year in the hatchery, taking up space, labor, and resources. So the motivation for my project was to assist in Pinto abalone hatchery production. I talked with Josh Bauma, who runs the hatchery for Puget Sound Restoration Fund, and he had suggested that he wanted to know more about how to support post-larvae because they are vulnerable at that stage. He was experiencing a lot of mortality and slow growth, and that he had been relying on nutritional information from other species of abalone, but wasn't sure if it was relevant to Pinto abalone because other species are farmed extensively in Asia, but they grow very quickly and they live in warmer water than Pinto abalones. So what they eat or what diatoms work for them might not be suitable for these abalones or for colder water. To give you context for the size of abalones I was working with, these red marks are numbers that I drew with a Sharpie marker. And this is a post larva when I started my experiment a week in. And this is 8 weeks later, it was about the size of a poppy seed. The benthic diatom diet. Abalones are face down licking rocks. So diatoms, what are diatoms? You're probably most familiar with them as planktonic diatoms that float in the water. Diatoms are single-celled algae with a glass shell wall called a frustule. And you can filter them out with a plankton net and look at them under the microscope, and they're very interesting and beautiful. They also can be a hazard to human health. If certain species form blooms, and those species emit neurotoxins such as domoic acid, they can result in high concentrations of toxins in filter feeding shellfish like oysters and clams, and then a closure of those beaches for recreational harvest. I worked with benthic diatoms, however, which are diatoms that stick to surfaces. Specifically, they have stuck to the surface of my boat. This is where the anti-fouling paint was still effective and kept the diatoms from growing there. And here in the dark areas is all the slime. It's a little pixelated, there's not a lot of texture there, it's just a uniform film of dark diatom slime. This is my rendition of diatoms growing in toilet water that is clean water, but that you have a sunny bathroom and they can establish colonies. And then when the toilet flushes, it simply circulates water past them. And I found this the other day while working on a boat. This was under a graded shower floor in a customer's boat, and this will fill up with shower water, then be pumped out via this hole, that's a wire. So the bathroom was sunny and they were forming a film in there. So while diatoms might be a nuisance for us in their benthic form, they are food for post larvae. And the suitability of the food depends on the size of the diatoms relative to the mouth of the post larvae. The shape of the diatoms, again, how easily they can handle it and bring it in. How strong the shell is. If the diatom has a strong frustule, the post larvae might simply eat the entire thing whole and it passes through their gut intact. which they do get nutrition out of the extracellular mucus outside of the cell wall, but not as much nutrition as if they could break that frustule wall and ingest the contents as well as the outsides. So mucus can add food value if they digest the mucus on the outside of it. However, if it's high adhesion strength, it could be harder for the post larvae to loosen the diatoms from the surface they're feeding on. And then of course, nutrient and biochemical composition affects food value of diatoms. So diatoms do move. That's why they have so much mucus, is that they're gliding around on little slime trails, kind of like slugs. I'm going to play you back and forth of two images, and you can watch carefully the position of these individual diatom cells, the little dots there, as they move back and forth between the two pictures that I took. And this will move back and forth too, because it's an abalone crawling along. Yee. Oop. So these are the 6 diatom species that I fed in my experiment. I'm going to introduce them to you one by one, but I'd like to show you that I've color-coded them throughout the presentation. So you'll see these little snapshots of them on my results graphs, and you won't have to memorize their names. You can remember what their shape and color were. Achnanthes brevipes was a diatom I was interested in because it grows on a short mucus stalk, as you can see in the scanning electron microscope image. There weren't any formal feeding studies in the literature on post larvae less than 1 millimeter in shell length. My post larvae started at 400 micron shell length, or in other words, 4/10 of 1 millimeter, and ended slightly over 1 millimeter long. However, Ezo abalone and South African abalone can eat it, and Ezo abalone grows quickly when fed Achnanthes brevipes, so it seemed worth trying. Amphipora paludosa has twisted frustules that leave double tracts of mucus. The red abalone post-larvae experiments have shown that red abalone grow well on it, but otherwise it hasn't been in the literature. But I thought that the double tracts of mucus might provide more nutritional content, especially if my abalones were too small to ingest the whole diatom. And for this slide and the next 4 diatoms, this is what the film looked like through my microscope, and this is the edge of a post-larval shell for context. These aren't fantastic photos because I didn't have a very strong light microscope, but I wanted to give you a sense of what the film looked like. So this film is fairly thin and has clumps. This film has larger cells that are more uniformly distributed. So Amphora salina is already in use by the Puget Sound Restoration Fund. They chose it, I believe, because it is local to the Pinto abalone range. It was isolated, the culture that I got, from Saanich Inlet, British Columbia, which is about 50 miles from Anacortes, where I was doing my experiment. It's only been studied in the literature 2 other times. Cylindrotheca closterium forms a thin film, and the cells are smaller and not as dark and round as the other ones. It has a thin frustule. You can see that when I took a scanning electron microscope image, it actually broke during that process. My other algae species did not break during the process, so it wasn't just me breaking things. Survival in other abalones when fed Cylindrotheca closterium is typically above 70%, and growth is good or excellent. And towards the end of my experiment, when the algae film was thicker, it was really obvious for me to see that my abalones had been eating the edges here, and this is the part they hadn't gotten to eat yet. And this is just a scrape mark, but the abalones were leaving these round, cleared areas. Navicula incerta is a diatom that's available as a paste from reed mariculture, which could make it very convenient for Puget Sound Restoration Fund or other hatcheries to feed if they don't have much capacity for propagating their own algae, because you can just take a little bit of the concentrated feed and dilute it in water and feed it. The genus Navicula is commonly isolated from hatchery intake waters and other experiments on other species of abalones. And then it's just identified to genus level rather than specific species. It's very common, Navicula incerta the species, it's very common as a reference diet for abalone feeding studies. The results are usually moderate survival and growth, nothing spectacular, but I have a suspicion that it's an easy diatom to identify and cultivate, and that's why it's so popular. Nitzschia laevis is a smaller cell, and it has weaker attachment. It tended to form clumps in my study, but didn't attach very strongly to the containers that it grew in. So it should be easy for post larvae to ingest, even if they're very small post larvae. Performance of abalones fed Nitzschia laevis in other studies varies widely, but I believe the genus is used in bivalve hatcheries for feeding oysters, so it seemed interesting to look into for abalone. The experiment, you can see I found many creative ways of looking at my tiny, tiny abalones. This was not the most effective, but it was the most fun. So the feeding trial that I conducted, my questions were, does diatom diet affect post-larval survival? And if so, which diet supports highest survival? Does diatom diet affect post-larval growth? And if so, which diet supports fastest growth? Can a diatom's size or shape predict its success as a diet? Can a diatom's carbon-nitrogen ratio predict its success as a diet? So the experimental design of my feeding trial, I had 7 treatments, each of them with an initial sample size of 18 per treatment. 6 of them were those live diatoms that I showed you, and 1 of them was a starvation control. Remember that the post-larvae live on egg reserves while they're free swimming in the water column, and sometimes those egg reserves can last for several weeks after settlement, even without the abalone having anything to eat. So I wanted to be sure I understood how much death would occur if they were fed nothing at all, and how much growth would occur, compared with whatever they were fed in my experiment. So of the 6 diatoms, I measured diatom size, and at the end of the experiment, 1 week after I removed the abalones, I sampled the diatoms and analyzed their carbon to nitrogen ratio. I measured survival of the post larvae at days 20, 26, 38, 49, and 61, in other words, every couple weeks. I analyzed growth at days 7, 20, 42, and 61. These aren't the same days as I measured survival other than the final day. Because of the labor involved, we could only do survival counts 1 day or growth photos and measurements 1 day. And then I sacrificed abalone to examine their radula, which is their teeth, on days 5, 20, 33, and 61. The last third of my presentation is about the radula, so we'll get into that more if you're wondering what the heck it is. For the starvation control, I could measure survival at days 20 and 26. After that, they were all dead. And I measured growth at days 7 and 20. Again, when they're dead, I can't measure how big they are. I intended to take radula measurements from the starvation control, but due to some contamination that lowered my sample size and the high rate of death, I decided not to sacrifice any of those animals for radula and instead keep as much growth and survival data in my other part of my study as possible. Achnanthes brevipe, oh, sorry. So these again are the 6 diatom species that I fed. And the physical setup, I used 6 well plates, which are these clear plastic devices that are about the size of a postcard. They have 6 wells of seawater in them, and then I pipetted, I had assistance, 10 abalone per well into the plates. This is the incubator in which we grew them. These are signs reminding us that when we move the plates in order to change out the water 3 times a week, that we needed to move slowly to not splash any diatoms from one well to the other. These are spare abalones that I raised just for the heck of it, and they are still in the incubator. And this black plastic bag is covering the water that I prepared, the seawater that was filtered, UV sterilized, and pH buffered to 8. So the advantage of Zoom is that for those of you not interested in technical details of cultivation, the next 3 slides are about that, so you can sneak out and have a drink. I won't even notice. The experimental setup, there's 10 milliliters per well, which leads for good gas exchange at the surface of the well because I had no aeration or water flow. We changed the seawater 3 times per week using glass pipettes. We changed 80% of the water because we didn't want to scrape the bottom or risk damaging any other abalone or suck up too much algae with the pipette if we tried to take all the water out. An advantage of the well plates is that they are transparent, so escape is unlikely, or I can catch little buggers if they try to crawl off and go somewhere else to freedom. The downside of well plates is that I had to manipulate the abalones onto the bottom of the well plate to measure shell length. This is the random distribution of diets within the well plates. So every 7 wells across the plates had all 7 diets, including the control starvation treatment. And then within each of those 7 wells, they were randomly distributed. So they weren't always neighbors to each other, like it wasn't the same 2 species of diatoms neighboring each other all the time. 12 degrees Celsius was the rearing temperature for the abalone, with a 12 hours light and 12 hours darkness rotation. And the seawater was UV treated, filtered to 0.2 microns, and buffered to a pH of 8. Sorry. To start the experiment, I purchased diatom starters from the National Marine Algae and Microbiota Repository at the Bigelow Laboratory. I grew dense cultures of these algae in F/2 media and flasks in a separate incubator at 17 degrees Celsius and 16 hours light and 8 hours of darkness. That was to keep the algae growing at exponential growth, and no social distancing was involved. I thinned the cultures periodically to keep them dividing, because that's the most stable nutritional profile, is if they keep dividing. I added diatoms at 1 cell per square millimeter of the surface area of the wells that the abalones were in after the abalones went in those wells. On the larvae side of things, Puget Sound Restoration Fund gave us larvae, which we transported in glass jars to Shannon Point Marine Center. They acclimated in the incubator for about 24 hours, and then we pipetted again 10 larvae per well, and used GABA to induce the larvae to settle. GABA is a chemical that mimics the cues of crustose coralline algae, letting the post larvae know it's a nice place to settle down. And then we rinse the GABA, of course. Statistical analysis. Overarching themes of my data included repeated measures and unbalanced data, so I was coping with that when I chose my methods. For survival, I did a one-way ANOVA to test for overall effect of diet on survival, and then followed it with pairwise contrast to compare diets to each other. I had no a priori or beforehand hypotheses about which diet would be better than others, so I compared all of them to each other using a Tukey HSD test. Leaping down to this growth, I chose a linear mixed model because it can handle repeated measures very well. And the shell length, the model predicted shell length as a factor of diet, age, and diet-age interaction, and then a random factor of well, again, for those repeated measures of measuring the same wells each day, or each day we measured. I did double check that there was no effect of survival on growth. I used a linear mixed model to check that with age and survival predicting shell length, and there was no effect. The reason I did that was that I wanted to be sure that there was no advantage to abalones if half of their brothers and sisters died and gave them twice as much space and food. But there was no effect. They could all get along together with the densities in my wells that I chose, and they had plenty of food if they all shared. Yeah, sorry. And then carbon nitrogen ratio of the diatoms, I sent them to UC Davis to be analyzed, and then made qualitative relationship notations between the carbon nitrogen ratio and the success of the diets for the post-larval growth and survival. So the diatom results. Diatom had an effect on survival and growth. And this is a picture of one of my assistants, Delia, doing a water change in the cold room with the well plates out and the pipette that sucks out the dirty water and puts in the clean water. Of course, we change pipettes between those. So we found most death in the first 20 days after settlement. Unfortunately, I did not have the capacity to check whether or not the number of larvae we pipetted into each cell, so 100% of the larvae, actually made it through settlement and metamorphosis. Sometimes metamorphosis, things go wrong and the life does not continue of that larva. Or maybe they died after they settled, or perhaps they simply did not settle and then floated around as larvae until they perished. Nonetheless, Amphora salina clearly had higher survival than the other diets. There's a lot of statistical overlap. These are 90% confidence intervals. Sorry. Hey Dave, is the bottom of my screen covered by anything? I'm getting a weird warning on my computer. Like, can you see where it says error bars, percent 90% confidence interval?
There's something popping up and then going away.
Oh, that's annoying. Huh. I don't know what to do about that.
Please move this window away.
Yeah. Oh, it's funny. It shows that up on your screen. I don't have any capacity to move that window, so I don't know what to do. I will forge ahead, everyone. So Amphora salina, clearly the best survival. Achnanthes brevipes had very poor survival. Only one survived to the end of the experiment. And another way of looking at survival, this is a comparison of each of these diets and their day 20 in the light gray survival percentage, and then in the darker gray, their day 61% that survived. Again, the error bar is a 90% confidence interval, so there's a lot of overlap. So emphasizing again, Amphora salina, the local diatom, had the highest survival for Pinto Abalone post-larvae. And Achnanthes brevipes, not statistically different from the control starvation group, but in terms of the average number that died, it was lower. Once you've made it to day 20, if you're a post-larva, you have pretty good odds of surviving to day 61 if fed Amphora salina, Navicula incerta, Amphipora paludosa, or Cylindrotheca closterium. But if you are fed Nitzschia, laevis, or Achnanthes brevipes, you do not have as good of survival after day 20. Growth depends on the diatom diet that the abalones were fed. Again, Achnanthes brevipes that had a lot die also didn't have many grow, so there's very poor nutrition involved in those. Cylindrotheca closterium had highest growth in the experiment with a greater exponential curve than the other diatoms. And then the other diatoms had variable levels. For instance, Nitzschia laevis was lower until it spiked near the end of the experiment. However, many died, so this could be an artifact of the lower sample size being here. I fitted a linear mixed model growth curves to predict diatom diet more mathematically. And again, it showed Cylindrotheca closterium was the best diet with some statistically significant overlap between Navicula incerta, Amphipora paludosa, and Amphora salina. And then Nitzschia laevis was the worst performing in terms of survival. Carbon to nitrogen ratio did not seem to explain success of diets. So looking at survival, this was the best survival, Amphora salina, and the worst survival is here in the middle, although I had only one sample for this carbon to nitrogen ratio assessment. This is the second worst survival with Nitzschia laevis, and it's right in the middle of the carbon nitrogen ratio. And then the others are simply scattered. So it didn't explain survival. Growth, maybe if you are a little more generous, you can see that this one had lower carbon to nitrogen ratio. So more nitrogen is often more nutritious for the same amount of carbon. But the other diatoms don't particularly rank in any particular order for that. And again, the poorly performing ones are right in the middle. So in summary for my diatom winners, if you want your post-larvae to survive, I suggest that you feed them Amphora salina. Or if you don't have that, Navicula incerta works pretty well too. If you want your post-larvae to grow, feed them Cylindrotheca closterium. Or if you don't have that, Amphipora paludosa is a second choice. If you wish for your abalones to die, feed them Achnanthes brevipes if you like to take a strong part in their death, or simply starve them if you would prefer to be lazy. And of course, Nitzschia laevis is kind of in the middle and mediocre. So what's up with Achnanthes brevipes that everything died for? An interesting thing about this diatom was that the houseboat's moving because a big boat just went by. Achnanthes brevipes is quote unquote advertised to look like this. This is what online sources tell me the morphology looks like. And I did see some of that from morphology. This is an image in my light microscope, and this is an electron microscope image showing those large, long rectangular cells. But I also had a lot, the majority of them by far were these smaller, rounder cells like I used in the little profile picture. And they form chains, which is what this is. The light microscope could only focus on one at a time, so that's why this is so blurry. I looked into whether I had contaminated the diatom mixture of Amphora salina that I got, but it came in 2 separate vials from the algae repository, so that seemed very unlikely that the 2 vials that I grew in 2 separate flasks throughout the duration of my diatom growth were, that they were being contaminated equally and by the same thing. I was wondering if there's stress involved in the diatom diets and the resulting shape of the Achnanthes brevipes. Were they going through excess cell division, getting smaller and smaller and stranger shapes? That seemed unlikely that they would change shape so much because of the rigid walls of diatoms. They separate by, they have 2 halves of the wall, one slips into the other, and then they separate and they grow a new half of the wall that slips into the old half. So they have to maintain the same shape they are because they're glass. They are not like amoebas where they can separate and pinch off what's there and then be whatever shape they turn out to be. So I was wondering whether maybe it was sexual reproduction instead of cell division that was causing this unique shape, but I never did find an answer. I was wondering if they were toxic or if they were yucky, because even though the small cells should be easier for my post larvae to eat, the Pinto Abalone post larvae died and I literally saw them crawling across Achnanthes brevipes without eating it. They simply refused to eat it. Ezo abalone from Japan don't die when they're fed Achnanthes brevipes, and I was wondering if that's because maybe they can just preferentially eat only the large cells, or the small cells are toxic, and so if they eat the large cells, they aren't toxic, whereas my Pinto abalone perhaps were too small to eat the large cells. So it remains a mystery. Next, I'm going to tell you about the radula. The radula are abalone teeth. This is a dissected out set of abalone teeth that I imaged on a scanning electron microscope. And the reason I started looking into the radula, aside from making super cool photographs, oops, I mean images, don't tell Charlie Wandler I said photographs. There's no photons in an electron microscope. So I was interested in whether the diatom diet and its interaction with the teeth could somehow predict the success of the diet. So an introduction to radula in general. Snails have them, that's how they eat. This is a snail of a different species in an aquarium tank, and it is grazing on the wall of the tank, so the glass is facing you. And this is its radula coming out of its mouth and licking food off of the surface of the glass. In other words, abalones eat by being face down and licking the floor with a raspy tongue similar to like what a cat's tongue must feel like. It's not technically a tongue, but we won't get into anatomical terminology here. And the radula in a Pinto abalone post larva is a little different to see, but you can see it right through the head because the head is translucent. It is this small zipper-like appearance here. And I was curious about radula tooth plasticity. So just like dinosaurs, abalones develop new teeth throughout their lifetime and discard the old, worn out teeth. And so it's just kind of a conveyor belt of new teeth coming in and old ones falling off. The advantage of that is that if you're making new teeth, well, you could make them to fit whatever food you're eating if your food changes. And this does happen in other species of gastropods that have radula. This is a species that eats macroalgae. These are adult teeth. And in the exposed habitat, where the algae undergoes a lot of wave action and becomes very tough, the teeth of the snails that eat that algae are serrated, and here they're smooth, where they eat the tender seaweed that doesn't have to grow as strong because the habitat is sheltered. So that's one example of morphological plasticity of teeth in response to food. It can be due to either chemical cues or physical cues. That's been studied in other species and families of snails. And I thought that if we understood the radula food interaction better between my abalone teeth and what they were eating and whether they were surviving and growing, it could help inform hatcheries' decisions about what to feed. So my questions about the radula were, are Pinto abalone radulas different from other abalone radulas? If abalone survive well, is it because teeth match diatoms? Is radula morphology plastic in response to diatoms? So radula preparation, I dissected radula out of abalone, which is a glorified way of saying I dissolved the abalone with bleach until the soft tissue all went away, and the only thing left was the shell and the radula. So again, here's where the radula is located, and that's what it looks like when it has come free of the shell and it's floating around. Then I rinsed it several times with a pipette, 4 to 6 times with fresh water to get rid of any salt crystals or bleach, and let it dry with the teeth facing down. For scale of what I'm working with, this was an American dime, and this is the radula curled up on the side of it. I imaged the radulas using a scanning electron microscope. These are the equivalent of microscope slides for electron microscopes. We put pieces of double stick tape on them, specifically carbon tape, which works for electron microscopes, and then touched it to the abalone radula that I dried, and then coat it with gold palladium so they show up better with the electrons bouncing off, and take an image with the electron microscope. This whole thing here, including the part that looks like a cabinet, is the electron microscope. The samples go in here, and then I controlled it with this computer. So giving you a basic introduction to radula anatomy in abalones, the Richidian tooth is a single tooth in the middle. It is symmetrical. It's always there in every row. There are lateral teeth on either side of it, which are also larger teeth, also used for scraping and digging. And as the abalone ages, they get more lateral teeth, and they're just numbered from the inside to the outside. And then marginal teeth are delicate teeth on the outside that work to sweep food rather than scratch and dig at it. And the next few slides go over the development of the radula sort of qualitatively throughout my study. So the teeth are very delicate and highly serrated, and in 5-day-old, there are only about 2 marginal teeth on either side, and usually 1 lateral tooth on either side of the Richidian. Sometimes the division between marginal and lateral can be hard. You're kind of guessing whether it counts as one or the other. And 17 rows at day 5. At day 12, they've added a couple of rows in the total length. Again, the teeth are still very finely serrated, and they have 2 lateral teeth on either side and 3 or 4 marginal teeth on either side. At day 12, this is a comparison of the different diets they were eating. Definitely no difference yet. At day 20, again, they added a few rows and then added some marginal teeth, but not a lot of change. Day 33, the marginal teeth are increasing much more rapidly now. The lateral teeth remain the same number, and again, a slow increase in number of rows. Day 61 post-settlement, the biggest change again is the number of marginal teeth, and then some of the abalones have 3 lateral teeth on either side and some have 2, so they're kind of in the middle. And for fun, I got a juvenile from Puget Sound Restoration Fund and dissected it for its radula, and you can see a large change here, and that's related to the fact that they're eating some more macroalgae now and not as much reliant on the diatoms. So the Richidian tooth in the middle is still present, of course, but it's gotten more blunt, and it looks like the tip points directly towards the viewer, whereas these lateral teeth, like the younger teeth in the abalones I just showed you, have the tips pointing kind of sideways to the viewer, which means that they might scrape the substrate more like a snow shovel that scoops up loose materials rather than like a hoe digging in a garden and loosening what you want to eat. There are way more marginal teeth in the juvenile radula, and they are very fine and sweeping the substrate on the sides. For comparison with other abalones, this is the development of the Ezo abalone, which I've mentioned a few times, from Japan. And they both start with very finely serrated teeth and not very many marginals. And you can see that with time, the Ezo abalone is developing a lot faster, especially if you compare 61 days and 63 days. The Ezo abalone, those middle 5 teeth are already looking a lot more like the Pinto abalone juveniles were, where they're ready to start scratching at the surface instead of just scooping loose materials up. However, if you consider size rather than age of the the radulas, we see that they scale pretty equally. The development is more dependent on size of the radula than on the age of the abalone. So 5-day pinto, 9-day pinto, they're getting bigger, but the morphology is still quite similar. Ezo abalone at 6 days is pretty equivalent to pinto abalone up to 33 days. At 61 days, the pinto abalone is more developed than the 6-day Ezo abalone, but it's pretty comparable to a 17-day-old Ezo abalone, and the size slightly smaller here. And then the 49-day-old and the 63-day-old Ezo abalone are much larger, and that would account for the fact that their teeth are more highly developed, trying to start eating tougher foodstuffs than just loose diatoms. So, is the pinto abalone radula big enough for my diatoms? Yes, just barely. Remember, the radula width is approximately the width of the mouth. And so if a diatom is wider than the mouth, wider than the radula, it'll be hard for the abalone to eat it. And then at day 30, they are definitely small enough. The radula was definitely big enough to eat the smaller diatoms. Cylindrotheca closterium might be a little wider than the mouth of the younger 12-day-old radula, but again, that was a very breakable diatom, so it should be accessible to the post-larvae of pinto abalone to eat. I'm going to let someone in from the waiting room, maybe. Nope. Okay. So I took more quantitative radula measurements than just counting the teeth in each row. I measured the number of fully formed teeth from the end of the buccal cartilage here, which goes over the odontophore, which is the structure that supports the radula, kind of like the inside of your tongue supports your taste buds. So I counted the number anterior to the end of the buccal cartilages to see what all was supported by that versus what was more rolled up inside of the head serving as standby teeth waiting to be deployed when the ones on the anterior side wore out. And I counted the number of fully formed teeth in each row. I didn't count these ones that were not fully formed and still under development, so I would have started counting here. I measured the radula width and the radula length following the center line, even if the radula was curved in the image. I've already told you about the tooth types, and I counted number of laterals and marginals. I'll show you statistics on those. And I measured the gap length, or the distance from one row to another. And that tells me whether, as the abalones, as the radula gets longer, is it getting longer because it's adding a bunch more teeth, or is each row of teeth getting further apart, thereby increasing the length of the full abalone, of the full radula? To try and get that snow shovel versus hoe effect quantitated, I measured the Richidian tip length and the Richidian height with the idea of, for the same height of Richidian, the more of the tip you see, the flatter the contact angle is, so the more that the tooth is scooping up like a snow shovel rather than digging into the food like a hoe. So I found that the radula scales up linearly with shell length. These are all shell lengths of abalone, but they're all 61 days old. I found that radula length increased faster than radula width. So like you saw in that juvenile post-larva, the juvenile radula, that was getting really long and skinny looking because it gets longer rather than wider as it grows. The distance between rows increased, but the number of rows did not. So although the number of rows was increasing with the age of post-larvae, within any given age, it looks like they had approximately the same number of rows. And the distance of rows was correlated with shell length, so that the bigger the shell, the bigger apart those rows were, even if they were the same number. The Richidian height and Richidian tip length both increased linearly with shell length, and they were in direct proportion to each other, although it was a very loose relationship. So they pretty much stayed the same shape from this metric. Marginal teeth, as we discussed, increased much faster with growth, and lateral teeth increased more slowly. So same with growth as with age. The Richidian landmarks. So in addition to those measurements, I did a couple other ways of determining radula shape. One of them was landmark analysis. So On every radula that I dissected and had a good image of, I took one row and I marked the dots where the widest part of the Richidian tooth was, the tip of the Richidian tooth, the narrowest part, and the widest part at the top of the Richidian tooth. And then also for a row, I used the 5 center teeth and got their top triangles measured. Not measured, geometric coordinates for it. And those coordinates are saved as XY coordinates, kind of like you might do with a map. And then I compared their positions depending on what the abalone was eating or how old it was. And I also traced outlines of the radula for the center Richidian tooth and then lateral 2 on either side of it, and compared those outlines to each other. I had wished to do statistical analysis of the outlines, but I found I did not have that capacity if it exists in the world. But I overlaid, I scaled them all, so some were bigger, some were smaller, but I scaled them all to the same approximate scale, and then using a statistical best fit kind of thing. And I don't see any obvious pattern differences. It's not like Amphora salina has a really short Richidian tooth and one of them has a long, deeper one, tooth tip, or same with the laterals aren't further out on some or further in than the others. So no apparent lack of plasticity in the radula and lateral 2 outlines, Richidian and lateral 2, excuse me. Landmark tooth shape. The gray dots are just my artist rendition of where the landmarks are for reference. And then the colored dots correspond to the average of all abalone at that age and where the average of their position of that landmark was. So the fact that these dots are clustered together indicates that there's very little difference between the ages of abalones. And so the tooth shape did not change with age in the days of my experiment to day 61 post-settlement. Likewise, I found no difference in diet in terms of the landmarks of the teeth. So the turquoise dots are the positions of the landmarks for all the teeth of abalones that were fed Cylindrotheca closterium. And the tips of the arrows are the positions of the landmarks for the abalones fed Nitzschia laevis. So you can see a few of these arrows moved very short distance. Some of these arrows, you see 2 things poking out of the dot instead, and that is actually the back side of the barb of the arrow. So the tip of the arrow is directly under the dot. In other words, very, very much not any difference. So in conclusion, diatom diet makes a difference to the success of post-larvae, Pinto post-larvae. Amphora salina was the best diet for earliest survival. Cylindrotheca closterium was great for growth, especially a little later in my experiment. And then Achnanthes brevipes appears to be a death sentence for Pinto abalone post-larvae. Pinto abalone post-larval radula is similar to other abalone species. It is driven by size rather than age. So if you are comparing or trying to make inferences about the radula and basing those inferences on other species, look at what size the animals were when they were studied, not the age they were. Why do some diatoms rock and some diatoms suck? I don't have answers to that, but I encourage future workers to look into it. I wish that I had been able to do more biochemical analysis of my diets. And also, I had more awareness now than I did at the start of my experiment about just how much the culture conditions will change how well diatoms perform as a diet. So culture conditions can really heavily influence the nutritional content of diatoms. And then also so does the specific strain of the diatom species. So one study might use Achnanthes brevipes, but it's a different strain than what I used. And so that could explain differences, for example. Are the same diets best for older Pinto post larvae? That's a question I encourage future workers to ask. I terminated my experiment at 61 days because of basically practicality of doing a master's experiment. And I encourage future work to extend that further into the post-larval stage for Pinto abalone. Will diatom pairs work twice as well? In other words, if we feed Amphora salina for survival and Cylindrotheca closterium for growth, will we get all of the benefits of each, or will there be contraindications? Some diatoms have allelopathic compounds that try and kill other competing diatoms or other competing algae. I did find information showing that Cylindrotheca closterium has some allelopathic compounds against non-diatom algae, but not against diatoms particularly. And then of course, even without allelopathic compounds trying to kill each other, the diatoms have to both have space and resources, and one can't simply grow too big and take over the other's space and resources. And what can we infer from other abalone species? Again, I noticed that with the Radula, we should be basing that on size rather than on age. And I would say that that is probably true for the success of the diets as well, that we should compare sizes more so than ages. So I'd like to acknowledge the funding I received from the National Shellfisheries Association in the form of a Michael Castagna student grant. The Pacific Northwest Shell Club gave me a scholarship, and some of their members are here watching today, and I thank you for that. The Washington Department of Fish and Wildlife gave some funding for Puget Sound Restoration Fund, that in turn I got some funding for my project. Puget Sound Restoration Fund was instrumental for the inspiration of this project, and then also the, not just the inspiration, but the practical advice on how to raise diatoms and abalones, and they gave me diatoms as well. Miller & Miller Boatyard is my current employer and keeps me happy, healthy, sane, and paid. My then undergraduate, now graduated and on to successful careers, assistants were Delia Dolan, Bethany Willis, and Zach Mangus. I'd like to thank Shannon Point Marine Center staff, particularly Jean McKean, Kat Shue, Brady Olson, Kelly Bright, and Carrie Fredrickson. Scientific and technical services, Charlie Wandler and Dan Carnevale showed me how to use the electron microscope and supported my work on that. I'd like to thank my committee, Suzanne Strom, Brian Bingham, and my advisor, Deb Donovan. My graduate student peers that supported me and let us all bang our heads against the walls together. My family, and Dave Linger and his cat, who fortunately did not jump on me, either Dave or the cat. Thank you. I can't hear anyone. I don't know where Deb is. I could look at questions now.
Can you hear?
Yeah, hi.
I unmuted myself.
Oh,
sorry,
great.
Deb.
Excellent.
All right. So actually, I'll just now invite questions for Lillian. We've got a few minutes for questions.
Should I start with the ones that people typed to me?
Sure. Why don't you do that?
Okay. So Madison asked whether Achnanthes of brevipes worsens the outcome compared with starvation. Like, does it actually inhibit survivability rather than just not being able to eat? Statistically, there was no difference between starvation and Achnanthes brevipes, so I think it was just that it was unable to eat and therefore starved to death. Carl asked, you mentioned that the wild populations are suffering from low population density. Are there indications that lack of food is a problem? I don't believe there are indications. I'm not really in ecology, so I'm not familiar with that, but I think it's the low population density of the adults leads to lack of larvae in the water and therefore lack of post-larvae settling. I don't think it's any problem with food availability for the post-larvae, certainly, and I don't think for the adults either. If Josh Bauma or anybody from Puget Sound Restoration Fund wants to unmute themselves and field any of the ecology questions, I'm open for that. Everybody's still typing, oh my goodness. So I think I touched on Madison's other question about mixing diatoms. Ben Fisher asked if there's a difference in how difficult it is to maintain the different diatom colonies that might also inform which diatoms should be used for hatcheries. I didn't find any difference in how easy it was for me to maintain diatoms. I did note some of them grew slightly faster in the culture plates than others. I actually thinned out all of the culture plates at about day 45 because I didn't want the abalones to be overwhelmed by this giant forest of diatoms. And then, yeah, they all reestablished themselves pretty well after the thinning. Marina asks, what did I spend the most time on that got the least amount of space in my thesis? That would be the radula shape morphology. Like, I was super excited to trace all these outlines and compare the curves, and I was going to see like how serrated one was versus the other, and if it succeeded. And then I found out that I had no way to measure like if it has like 3 serrations versus 6. Like it just wasn't tracking, and I couldn't do any analysis on that. Todd asked if all of these diatoms are commonly present in any given saltwater locale. I don't believe so. Most of my diatoms, I think 4 out of the 6 were isolated from Woods Hole, Massachusetts, and I think 1 came from the Sargasso Sea out in the Atlantic Ocean, and then there was the 1 local one. Maybe 1 was from like California. Unfortunately, I looked into at the start of my experiment what diatoms are native to Puget Sound, and all I could find was information on planktonic diatoms, and I really found nothing telling me about benthic diatoms, which was kind of disappointing, but I just kind of went with the aquaculture literature instead of whatever native diatoms were available. Cool. Todd asked if the radula is a single rigid structure or can the critter manipulate parts of it? It is not a rigid structure. It is on a flexible surface and it articulates with the joints between the rows, and so it scrapes stuff down. David and Julia asked if I kept a record of total hours spent on the project. Yes, I did. At one point I was feeling spiteful and that I wanted to quit if I got more than a year of full-time work in the project. I think it ended up a full-time equivalent of like 9 months or so. But I work pretty slow and I'm kind of perfectionist about certain things and I go down rabbit holes, so there's no one to blame but myself. If people want to ask questions verbally, that sounds nice now if your computer monitor stuff works.
So Lily, is any of this relevant to the impacts of ocean acidification and warming?
I am not sure. Like other animals with calcium-based shells, abalones are certainly affected by ocean acidification. And certainly if we can raise healthier, more robust abalones in hatcheries, then we should do that and they'll be more fit. But that's as far as I can go into that. I really didn't study ocean acidification.
Thank you.
Brilliant. This is Bob Lane. Great,
Hi, Bob.
great presentation. I wondered if diatoms have similar teeth structures as they move out, similar to sharks.
So
I'm sorry, not diatoms, but the abalone. Excuse me.
The abalone, the teeth structures change throughout time as the abalone grows and starts to eat other food. And yes, like sharks, they grow more teeth and then they shed more teeth and they have multiple rows working at once. Did that answer the question, or am I not quite understanding?
I'm learning so much. So thank you. It was a great presentation.
Thank you.
Hey, Lily, can you hear me?
Uh-huh.
Oh, hello. Okay. So I was curious, you know, the radula and the mouth are the same size, and you weren't able to detect any changes in morphology or changes in the size of the radula and the mouth. I was just wondering, even though you suggested that you don't think that the endangerment to that species sustainment is because of lack of food, but rather lack of opportunity for good sex. But does that mean that there was fewer diatoms that you could experiment with because they had to be small?
That's a good question. To be honest, when I looked into diatoms, I approached it from the point of abalone aquaculture, and then the diatom sizes, I kind of narrowed it down to diatoms that had been used in abalone aquaculture in general. And then when I chose my specific diatoms, I made sure that they were small enough for the abalones that I'd be eating, kind of based off of what I did know about the shell size of Pinto abalone compared with the shell size of other abalones. So
Yeah,
yeah,
that's sensible. You had to use what you could get for one thing, and what would be employed, you know, most likely, you know. Hi. Are you on a houseboat, Lily?
yes. So I think that the benthic diatoms in general, I think that the upper size range of them is not too distant from the upper size range of what abalone can eat. I'm not 100% sure on that, but I think it's true.
So at this point, I'm going to interject here because I can't see how many more questions there are, but Lillian does have another big event coming up in about 20 minutes. And so I do want to make sure she has some time to decompress a little bit and get ready for her defense. So I'd like to just thank Lillian for a great presentation, and let's give her a round of applause. I hope she can hear it.
Yay, Lillian.
And thank you everybody for coming. And she now has a list of all of these different questions, so she can get back to you on a one-on-one basis. Maybe not this afternoon, because hopefully she'll be decompressing after her her defense.
My boyfriend promised to spray me with champagne if I pass my defense.
Okay. Hopefully he has a bottle ready. All right, so thank you, Lillian, and we'll see you again in about 20 minutes.
Okay, thanks everybody.
Good job, love.
Bye. Good job.
Bye.
Bye, Bud. Bye, Andy.